думают, что мы спортсмены. За мной! Открывай дорогу, дядя! Физкульт, привет, дядя! Салям алейкум!
Вам что, товарищ?
Салли, солнышко, это на планерку. Проходите, товарищи. Спасибо, ребятушки. Сегодня у нас на повестке дня строительство домиков для заключенных вместо бараков. Располагайтесь поудобнее, записывайте, запоминайте. Государственная программа по постройке жилья касается всех. Начну, пожалуй. Внимание.
Домики, домики, как грибы растут, знаю, новоселья все, конечно, ждут. Домики, домики, радуйся, семья, каждому по домику обещаю я. Домики есть у всех, точно вам скажу, даже и у глупенькой антилопы гну. Домики, домики есть у соловья, и заметь у каждого, даже муравья. Ай да, ребятушки, в лес не по дрова, с рук бы, ребятушки, нам рубить пора. Надо управиться срок нам до зимы. Ай да, ребятушки, вместе сила мы. Домики, домики, как грибы растут, знаю, новоселья все, конечно, ждут. Домики, домики, радуйся, семья, каждому по домику обещаю я. Много листов у нас, много и земли, жизнь она не вечная, сам ты рассуди. Домики, домики, как грибы растут, надо не на небе жить, и надо жить там тут. Ай да, ребятушки, в лес не по дрова, с трубы, ребятушки, нам рубить пора. Надо управиться срок нам до зимы. Ай да, ребятушки, вместе сила мы. Домики, домики, как грибы растут, знаю, новоселья все, конечно, ждут. Домики, домики, радуйся, семья, каждому по домику обещаю я. Крутится, вертится в космосе земля, знаю, все изменится скоро у тебя. Домики, домики, как грибы растут, надо не на звездах жить, надо жить нам тут. Ай да, ребятушки, в лес не по дрова, с трубы, ребятушки, нам рубить пора. Надо управиться срок нам до зимы. Ай да, ребятушки, вместе сила мы.
Спасибо, ребятушки. Можно приступать к работе. Работать будем быстро, но аккуратно. И не забывайте про технику безопасности. Всем хорошего дня.
Теперь не мелочь, по карманам тырить. Ну пусть я чистить кирпичи класть.
Давай, червонец, пожалуйста. Открой кидать.
nobody paying attention. It's okay, it's okay.
sa bouche qui devient dure. À voir ses yeux, je crains le pire. Je sais que bientôt je vais souffrir.
Hi, good evening, everyone. So we're gonna start the talks. A short introduction. I don't know if you can maybe shut down the music. Thanks. Just out of curiosity, who is coming the first time for a data science meetup? Please raise your hand if it's the first time that you come at a data science meetup. Okay, so we have pretty around 5-10% new people. So actually, the data science community in Brussels, it's already the 5th season. We start in October this season, and basically, as you can see, there is multiple initiative that's going on in Belgium. There is meetup in Leuven. There is actually today the first data science meetup in Hasselt. And there was also in Liège, Ghent, Antwerp and Liège. I don't know if they are that much active at the moment. Liège is active, okay. But so you have a view. It's not only in Brussels. Okay. So this is just the spirit. We always try to pick a team. So today is around health. We did the first one on sports, and we will have other meetups on team on legal and also legal tech, arts. Yeah. So if you know people involved in data and arts or data in legal, please come and talk to us. So this is basically the theme of this 5th season, with of course the big boss over here. And that's pretty much it. So today the agenda, we have 3 nice speakers coming from different area. So we have more startup area, UCB, I think you mostly know. And also Tony, which is a freelancer working as a consultant. A small word about a hackathon that happened last weekend. I'll give, I think Philippe will gonna give a few words because I think it's worth it, as the team was also around health, right?
Okay, well, a very, very warm welcome to all of you. And for those who are for the first time here at Digitizer, know that this is the house of the data science community. This whole place would not be here without the data science community, which we started nearly five years ago, and that we build up to about 3,000 members today. And since we started, we've done 170 events of all different sorts, and we're very proud of that, very proud of the support of all of you. And it allowed us also to train a lot of new people into data science. So we started the Data Science Bootcamp. We are now in the 3rd season of the Data Science Bootcamp. So who in this room is an ex-bootcamper? I see William. Yeah, yeah, there are about 5, 6, 7. Who is currently a bootcamper? Yeah, these guys are looking for a job. So they're now in their 5th month of training. Basically, they've done the first 8 weeks of certification into R and Python, and then they went over into doing business cases. So they've done about 15 business cases from A to Z, starting from scratch and defining until the end of the solution. So very warm welcome. What we also did together with the boot campers was a Data for Good hackathon. We like to do hackathons, but we prefer to do it for a good purpose, to change the world, to become a better place. 2 years ago, we did a hackathon on dengue, which is a tropical disease virus, where the virus is mosquito-borne. This year, we decided to do it on HIV, and specifically on HIV drug resistance. We had on Friday 107 people in this room. Warming up for the hackathon. We started at lunchtime. By the evening, we had 15 solutions that were already sketched that the people were going to work on. And by Saturday night, out of those 15 solutions, we kept 11 solutions. This year, with this Data for Good hackathon, we want to go one step further. Normally, we stop at the delivery of the solutions, which like, okay, now you've been in contact with VPs from Janssens, it's up to you, you know. This year we want to go one step further. We want to deliver part of the solution in one of the countries. The whole topic was more because it was HIV drug resistance. There was a lot of there's a lot activity around that going on in Africa. And what we wanted to do is to create a model that we then would export to one of the countries. We would then teach the local data scientists how to use our model, and they would use their data with our model. And we don't touch their data. We just say, okay, this is the model. We'll teach you how to use it. It will give you a visualization on the important areas in your country where you need to communicate better. But we're not touching your data anymore. We teach you how to fish. And we're not throwing fishes at your head. So that's being said for the data science community. This place, we used to be at AXA. Who remember us at AXA? Yeah, familiar faces. We were very lucky because AXA allowed us to do the first Data Innovation Summit, to do the second Data Innovation Summit, each time with 500 people, and also gave us 500 square meter or even 600 square meters of office space and 12 training rooms. Because we had 12 training rooms, we then said, let's do trainings. And so people like you said, oh, I have something to share, and I'm going to share my knowledge with data scientists. I can decide myself if I want to ask money for it or if I want to do it for free. And we did that. We started that at AXA. We're continuing doing it here as well. So if you have a topic that you want to share, we will organize the event with you. We will make sure that there are people in the room that will listen to your topic. If you don't ask any money for it, the event will be for free. If you ask money for it, we will promote that as well. And we will keep, as a community, 30% for as an organization fee and rental fee for the office space. Yeah, here you have about 2,000 square meter of office space. You have a very nice rooftop. I really before you go home tonight, go on the rooftop. It's new. We rebuilt it this summer. On that rooftop, there is a training room where you can put 60 people in it. There is an eating space where you can eat with your team and then and benefit from the terrace. It's being used a lot by bigger corporates today because their day off or their monthly meeting, they prefer to do that off-site near a metro station, and we are very near to the you cannot be near to the metro station as we are here at, because the exit is just in front of the door. So this being said, I think it's high time to announce the next speaker. So I will ask Mehdi to do it because he is the president of the evening.
Thank you, Philippe. I think we can do a warm welcome for Tony, which is our first speaker. Thank you.
Good evening. Is this working properly? Yes. Okay, great. And that's my slide, so that's fine. Good evening. So my name is Tony Verbeeren, and I'm going to tell you the story of, that sounds better. I'm going to tell you the story of Compass, also known as Lucius. First, 2 words about myself, not too long, because you don't want to spend too much time on me. After my PhD in theoretical physics, I moved into IT consulting, infrastructure consulting first, later on services, IT services. I did a short postdoc at the University of Leuven, KU Leuven, and it's then that I started collaborating with people from Janssen Pharmaceutic. And it's then where this work, which I will talk about, all started. And from then, a little bit before that, I started as a freelance as well, and that's what I still do nowadays. Okay, preface. because it's the story, it's built up as a story, of course. First about apples and pears. Well, not really about apples and pears, but rather about diseases and compounds and how to compare them. Because that's basically what we would like to do. The landmark paper that defines this whole field, let's say, or method, so to say the least, is this one by Lamp and others. And already here you see in the title, Connecting Small Molecules, Genes and Diseases. So we really want to compare these things to each other. And the whole idea is based on one simple aspect. Let's get a fingerprint for a compound. Now there are several ways to do that. In chemics, people have done that for ages, let's say, but we want something specific. We want a fingerprint for a compound, and we need to be able to compare these fingerprints to calculate the distance, so to speak. It has to be biologically relevant. It has to connect to genes, to biology somehow. And so what we will do, we will look at the effect of compounds on genes. It's as simple as that. And so you connect compounds to genes. Now it sounds of course trivial, but it's not that simple. But the idea is that. So you do experiments, you test compounds on a number of genes, and you see what effect they have on each gene separately. And so you have a way to, basically you have a space representing these genes, and your compound can be mapped onto that space. Now, to make it a bit more simple, mainly for the experimental purposes, not a full gene set of human beings is used, luckily. I will come back to that later on. We have around 22,000 genes. It's reduced by means of a random forest model to 1,000 landmark genes. And those are the ones that are tested in the laboratory for every compound, for every sample that is put in. Okay? That's basically what happened at Janssen. So until now, we have around, well, a bit less than 500,000 samples, representing a bit less than 250,000 compounds. So some compounds are tested multiple times. One reason for that is we have two cell lines, two cell types to do those experiments in. And then we need our distance. And there are several ways to do that. People have looked at Tanimoto distance, for instance, for people who are familiar with that. We use a relatively simple way to calculate the distance between those vectors in the gene space, so to say. To do that, we first convert the vectors to ranked vectors. And then it's basically a dot product, a normalized dot product between those vectors. And that defines the distance that we will be using. So in practice, we did our experiments. We have our 500,000 samples, each with their vector of strength 1,000. And we have a query. For instance, we are interested in a certain disease, and we know this disease has a certain effect on genes. And so this gene list, basically, is our query. And we compare it, we calculate the distance, or the Zank score, or the similarity. It's all similar words, different words for the similar thing. We calculate the similarity with all the samples in our database. And then, well, the first thing you can do, of course, is create a histogram. A histogram of all the similarity scores. +1 means highly correlated, -1 means highly anti-correlated. You can also represent it in the so-called similarity plot. At first I was confused when I was looking at this. It's something that has been used by the people involved in this project. It's relatively simple, but you have to get your head around it. On the x-axis, you get the similarity scores, or at least the samples, ranked according to their similarity scores from high to low, from +1 to -1, left to right. On the y-axis, you have exactly the same. So why don't we get a straight line? Well, it's very simple. You add noise in the x direction. You basically add just random noise. And then what's the nice thing about it in a sense? Well, you get a two-dimensional graph rather than a histogram, but that's not a big deal. But you have a nice idea of what are the top correlated and the bottom correlated samples. And that's the main reason why people have been using this. But because we will encounter the similarity plot later on again, I show it to you. Okay, so that was a bit the context. Now, to the introduction. Here on the slide you see Hans de Wolf, and he had a problem. You see it on the picture almost. He's a biologist at Janssen, and he got his first 1,000 samples from the experiments. And he was looking at them. And the IT people at Janssen, they were very supportive. They set up a typical database, a Spotfire database, sorry, with a Spotfire front end. And he could really look at this data and try to create, for instance, the similarity plot that I just showed you, based on a, let's say, random query. Now, that didn't really work well. It was way too slow, even for these 1,000 samples, and it often crashed. And so he thought, well, if this is 1,000 samples, soon we will have 500,000 samples. Well, that was 2 years after, but still, they were in process of being done, generated. So that was his main worry, let's say. And that's when we met, and then that's when I started to think about possible solutions to that. And the first thing we did was distributing the computations. Because you need to calculate the distance or similarity with every sample in the database, you can very easily distribute that. There's no links or relations between any of these, let's say, baskets in which you put those things. We use Spark for that. Who is familiar with Spark? Raise of hands. Okay. We used Spark prior to version 1. So that was really, let's say, early adopter stage. And it did really what we wanted it to do, at least, especially in this scenario. So we started developing some IPython notebooks. Back then it was still called IPython, now it's Jupyter. And we were quite easily able to recreate the similarity plot. And so we said, oh, okay, this is nice. Let's make it a bit more fancy, work on the graphics a bit, even interactive plots in the notebook, all feasible, of course. And then this is kind of the latest stage where we abstracted away most of the functionality into a Python module so that people could just really look at the notebook, use functions and abstractions, and really easily do an analysis like this. Okay, so I was convinced that it was my endpoint, that I had delivered what I needed to deliver, at least in the project. Now, we did some extra add-ons, let's say. People were interested in rendering the chemical formulas based on the SMILES code, which is a representation of the chemical formula. And we worked on the graphics a little bit more. This is using Seaborn, for those who are familiar. And then that was basically the endpoint. Some clear benefits. It's really flexible. The sky's the limit. If you know a bit of Python, you can do whatever you want, plot whatever you want. It's really handy and nice. But there were quite some disadvantages as well, as you can imagine. Otherwise, my talk would have finished here. Data had to be loaded in a cluster, because at that moment already we had quite some data. You cannot afford to have a cluster per notebook or per user. And so that was kind of a tricky thing to handle. But also, at least a bit of programming language, Python knowledge is required in order to do these notebooks. Then some people who needed to work with this data, this analysis, were chemists, biologists, so we couldn't expect them to be able to program Python. So that clearly wasn't the end goal. So then Hans started bugging me with questions like, can we not create an app? I see these nice web interfaces on the internet. Can we not do something similar? And I said, oh, no. All sorts of questions popped up, like what with scaling? If it's tightly coupled, what will happen? Every user will especially need a cluster. It's not possible, it's not feasible. I hardly had any web programming experience. So also there, I said, oh, who is going to support this thing? I mean, there's nobody who's interested in supporting this kind of beast, let's say. And we were talking about potentially more than 100 users. Also that, if it would just have been 1 or 2, I mean, that would have been reasonable. But so this would have to scale at least a little bit, especially with the cluster in the background and Spark, etc. So I had these nightmares, and I basically said, no, please, no front-end development, please, for me. And I thought this was finished again, but it wasn't. But luckily, I got some help. I got some help from 2 people, mainly. Herculano Campos and Thomas Moerman. Some people may be familiar with Thomas Moerman. Maybe, no. Yeah. Herculano helped me with the front-end stuff. He's a designer and a front-end coder. And Thomas helped me with some other component, which I will show you shortly. But first, we didn't want this whole thing to be tightly coupled. We want a loosely coupled system where the cluster and Spark are running as a service. And so we needed some component. I didn't want to develop it myself. At that time, the only component that existed was Spark Job Server. Nowadays, there's some more options there, but at that point in time, it was only this. And one little drawback was that there was no Python support yet for Spark Job Server. There was only Scala support, or Java, but really I didn't want to delve into Java. So we started all over again, doing the notebooks, this time in Scala. Recreating similar graphs. This is one of the first prototypes using the Zeppelin built-in visualization. Who is familiar with Zeppelin? Just for my own interest. Okay, thanks. Later on, we evolved that a bit as well, also creating a core library with all the core functionality in it, similar to what we did before in Python, with the Python module. Also some improved graphics. We have some bubble graphs already because, well, it became too many points to plot on one graph. And we used Vegas for that, just to make it a bit more standard compliant at that time, let's say. So that was that. And so slowly, we developed the necessary functions, code, let's say, library to work on this Lucius API. The API for connecting to Spark, let's say. I'm not going into the details of how the Spark Job Server works. Oops, that's too fast. Oops. Yeah. If you're interested, please catch me later. I would be really happy to talk further about this. So we have Lucius API. Thomas created Surir, and Surir is a way to render the chemical structures based on a Smiles code. It's really a very simple, in a sense, REST API. You send it to Smiles code, it returns you the chemical structure as a PNG. And then Herculano created the web interface. Just to give you an idea of what we were dealing with, this is, at least if everything works properly, this is Surir working. So I give it a URL encoded Smiles code, which is a representation of a chemical structure, and it returns me a PNG rendition of this chemical structure. Similarly, the Lucius API is a REST API, so I can ask it, in this case, to return me some statistics about the data set, the samples in the database, let's say. It's just a test database. It's this first database that Hans was working with. There's only a bit more than 1,000 samples in this database. It's just for development and test purposes. So, but this is basically how these things are glued together, you could say. That's for later. And so this is all then combined into this first version of a web interface. And everyone was really happy. And I'm still convinced that Herculano did a really good job. He took some standards, at that time at least, we're talking about 3-4 years ago, and it worked well, at least in the beginning. You could zoom in into the graph and things like this, but there's of course a but coming, otherwise my talk would have been finished now, and it's not. It looked good, people were happy, there was minimal client-side overhead, so everything was really done on the cluster, all the processing on the cluster, and it's just the REST APIs that did all the work basically, and that returned all the relevant information to be drawn on the screen. Now, disadvantages, not from a programming standpoint or whatever, but there was really a mix of two different types of analysis you could do, and we call it workflows nowadays. Two different types of questions you could ask the data, let's say, and that was all in one interface. And from a UI point of view, it didn't really work well. People were confused. We ran into troubles with usage of Polymer. Nowadays, you don't hear too much about Polymer anymore. I mean, I don't. But also, and mainly so, the availability of Herculano became a problem. He was working at Intel when he started working with us, and Intel was part of the same ExaScience project I was involved in, and so that was how the collaboration worked. But then he started as a freelance, and things became really, really difficult then. Okay, so, oh my, what now? There were feature requests coming in. We had to do some changes to the UI because there's these two workflows coming together in one interface, which didn't really work well. So, well, we were back to the drawing board, basically. And I was back with my same worry and nightmare, like, oh no, I don't have to do front-end development, do I? But in the end, yes. And then the first thing to consider is what kind of JavaScript framework will I use? I've been looking for a lot of memes on the internet about JavaScript frameworks. There's too many of them, so I didn't select one, none. I use Cycle.js. It's not really a framework even. It's really a small code base, but it allowed me to work in a functional way, in a reactive way, which is, to me, sounded, well, let's say, close to what I was used to. If you're interested in this whole thing, reactive programming, and how the creator of Cycle.js looks at these things, I advise you to look at this presentation. Another thing I chose was to use, remember we used Vegas in Zeppelin for the rendering of the graphics. Well, Vegas is based on Vega, which comes from the lab of Jeff Leake, who is a professor doing visualization research. One of the tools they have is also Vega. It's rather a spec, a JSON spec you can use to create declaratively your visualizations. And this was really close to what I had in mind, namely, you have this component, which is declarative, self-contained, and this is all you need in principle to generate this graph. Yeah. And so I wanted to use this. And then we needed different workflows, different types of analysis. There's currently 3 implemented. The 4th is foreseen soon. Let me go into 2 of those. Let's start with disease. You have a disease which you want to study. You want to look for drugs that are or at least compounds that are potential drugs for this disease. If you study this disease, usually you know, or you can search in the literature, or you do experiments to pinpoint what genes are involved in this disease, in this effect. And so, if you query these genes, you can see which compounds highly correlate with this list of genes, or are anti-correlated with this list of genes. I will immediately show you an example in real life. Similarly, for a compound, you're interested in a biological way which compounds are related to which other compounds. So you start with one specific compound, you represent this as, from data in the database, you represent it as a vector in the gene space, and then you can calculate similarity with all the samples in the database. Okay, demo time. So this is then version 2 of Compass with the 3 workflows. Let me go into the compound workflow, and then I have my notes here because I couldn't remember. Tamibarotene or something. It's a drug used in people with leukemia. There's 3 samples in the database that correspond to this compound, to this drug, because it's a publicly known drug. Well, some of the things are a bit fuzzy. That's on purpose, unfortunately. Okay, so I have a list of my 3 samples, and I can calculate the similarity scores, the similarity plot at least, with all the samples in the database. I have similarly the histogram, which, if you remember well, corresponds to more or less the same information. And I have top tables, the highly correlated samples in the database, together with their chemical structure, also a bit fuzzy here, again on purpose, and some more annotations with respect to these compounds. I can then expand this table. I can export it as a TS3 or a CSV file. I mean, sky's the limit there, as you can imagine. So this is basically the functionality that is currently available. One of the later additions is additional annotations with respect to genes, because, or proteins at least, for which they code, so that people can easily look up certain genes that are there in the signature. So that's basically it. I mean, that's the application. Similarly, you can, as I mentioned, you can query for diseases. Let me quickly show you. This is again a signature related to some type of cancer, if I remember well. And again, you have similar information, but makes it possible for people at Janssen to explore the data really the way they couldn't do that before. Okay. Oh, yeah. So I had to put this in. So suddenly I became a full stack developer. I had never estimated to read one. Okay, so these were backup slides, just in case Murphy would be involved. Come on. It's a bit faster on my screen. Settings, okay. Okay, I'm not going to talk about data or infrastructure. Please, again, ask me if you're interested in some more finer details, because there's a few things I would really like to mention. So this I'm going to skip, not because it's not relevant. The future. It's always nice to think about the future. We can currently more or less calculate similarities between compounds and diseases. And so compare apples and pears in a sense. Now, if you think about adverse effects, for instance, you take a drug and it turns out to be toxic for your liver. That's of course something you want to avoid. But toxicity is also something you can express in terms of genes, what the effect is on genes. So again, we would be able to include this kind of information, at least for the types of toxicity we know, that are published or that have been known. We can include them in the application as well. And let's say now we have a list of possibly correlated compounds. We could immediately see whether some of those are toxic, yes or no. So that's one of the next steps. Another next step, we did some analytics, and other people in Janssen have also done some more analytics on the data itself. What we are now about to do, also together with Boris, who is here in front of the room, we're going to look at correlations, clustering, and both supervised and unsupervised, in order to learn something more about this space, let's say, and how compounds relate to each other, again, on a biological level, not on a chemical structure level or something. Also something which is for the very near future. First, some machine learning has been run to extrapolate the 1,000 genes back to the 22,000 genes using a machine learning model. So we have the full gene set again. That's one thing that has been done already last year. Another thing is there are experiments currently running on the full transcriptome, on the full set of genes. So also there we have nice reference points to see what data corresponds to what. And then an epilogue. I just wanted to note that somehow the common thread in this work, which has been going on for 4 years now, luckily not full time, is this one. We always start with notebooks. I mean, I always start with notebooks, even in cases where no cluster or Spark or whatever is involved. If it makes sense, I tend to create libraries or packages. In order, for instance, to add automated tests and things like this. You can go back. You can again prototype in the notebook. From the libraries and the packages, it's relatively easy with existing open source components to create services. That's what you saw with the job server thing. There are other solutions nowadays to that as well. And from services, it's, let's say, relatively easy to go to the applications, really to front-end applications. A recent package that has been open sourced by Microsoft, MML Spark, even allows you to spawn services from the notebooks themselves. So that's also quite interesting. I haven't been able to test it yet, but it seems quite promising as well. Okay, that's it for me. Thank you for listening. If there are questions, I'm glad to at least try to answer them. And otherwise, please talk to me later on tonight or later. Yes. Yeah. It's a very good question. So part of the question about business case for this kind of applications. Well, it's quite clear, and I mean, but it's a point I didn't touch upon. So it's a very good question that you asked. So the idea is, nowadays it takes on average 12 years to develop a new drug. It has become more and more expensive, and especially the early stages are quite expensive because you have to test a lot of possible compounds. It's the chemists who do the selection of compounds, so they have no clue about the biology of their chemistry, let's say. So the chemists do the compound selection, then they have to do a lot of tests, a lot of experiments before they enter human trials and things like this. So it takes 12 years, a lot of million euros or dollars, and using this application, we can relatively easily already do a filter during the start phases. And for instance, adding the toxicity is very important because that's one of the things that has been thrown out during human tests, but you don't want to test on humans if you can be almost sure beforehand that it will be toxic for the liver, for instance. And so that's really the basis of this of the business case in this scenario. Yeah. There are already a few papers out. There are no, of course, again, it takes on average 12 years, so it's not for tomorrow. So a new drug takes on average 12 years. So it's not, this has only been studied for 1 year. No,
one more question, please don't hesitate to talk about just you. Yeah, coming. So yeah.
I have a question about you use only 2 cell type to screen for the entire organism. Like if you want to screen for liver toxicity, if you don't have liver cells, it's a bit weird.
Okay, we're not going to look at liver toxicity for all the samples. Liver toxicity is something that this has been studied, and we know which genes are involved, irrespective of the cell types. So there's a different thing there. But you're right in the sense that we only have studies nowadays for those 2 cell lines, and that's really a relevant critic, let's say. Yeah.
Okay, thank you.
But just to add to that, it's already very, very expensive to do all those experiments. So that's the main reason why there's only 2, obviously.
Another last question.
Yeah, I was wondering, why would you target directly genes instead of enzymes and proteins? Like that would be the first approach, right? Both for for activity and toxicity.
That's a very good point, which I really can't answer.
Okay, thanks again, please, Tony.
Thank you.
So next speaker we have Nassim. The floor is yours.
Sure.
Merci. Wow. Looks a bit weird, huh? It's okay. Good evening, everyone. Thank you for inviting me. Thank you, everyone, for inviting me and for staying for this talk. So, this is working. There's a bigger screen there if you want to look at. Yeah, for me that's okay. There's a bigger screen for those who want to see there. So I'm going to talk about a project at work that started with that question on the top of the slide and this small data, probably one of the smallest data projects we have in the last few years. Does anybody have the answer? So that was the question. How do you fit a curve to this data? Which curve do you want to fit? Data scientists in the room? The red one? Nice. Simplicity is a good rule. Becomes razor. Anybody else? All big data. So tell me. Oh, that's a good question. So I'll bring a bit more context indeed to make that more interesting. So I'm working at UCB, so it's a Belgian mid-sized biopharma company, and we focus on making treatments for patients with severe diseases in neurology, immunology, and bone disease. Mid-size, so a bit less than 10,000 people, still a global company, and also it contains the whole chain of what a pharma company does, meaning starts from drug discovery, finding the molecules that are helpful, development, so understanding for which patients and in which situations they are the most useful and potent, manufacturing, these drugs need to be produced, and then finally sales, marketing, and even after they're on the market, monitoring how well they really work, which is called real-world evidence. It's called biopharma because it produces biologic drugs. So, very quick introduction. There are two big kind of drugs. The one kind is chemical drugs, or called small molecules. It's the one on the left, and is an example. So these are very small chemical compounds, very small number of atoms, which can be, the big advantage, they can be synthesized chemically, so relatively easy to produce. And so when you have that's pretty amazing. And when you think about it, these little compounds are able, these are able to treat you, is pretty amazing because they can bind to what will fix your body, and they don't bind to what will be toxic, right? And when you think about it, it's so small, right? So the number of possibilities to bind can end up being gigantic, and then it will destroy everything on its way, and it's not useful. Or it will not act at all because it's just already saturated. And the fact that some of these can really heal people is pretty crazy. And so one of part of this research in that area is really to try as many possible compounds as possible. You can synthesize them, you can try them against a lot of things, and so you get this kind of large-scale analysis like the talk before, which is extremely interesting. And another route to go is to use the immune systems of humans or of mammals typically to produce antibodies. So antibodies are much more complex molecules that the body produces to target something that's a strange element in our body or something that needs to be destroyed. And so your body constantly, its immune system constantly is looking for foreign bodies and is building antibodies that are really targeted towards that. So it's really mechanism. And so these molecules are much more complex, typically thousands of times bigger than the small ones. You cannot synthesize them chemically, right? So you cannot bring different chemicals and just build it. The way to build this is through the advances of bioengineering, which started in the 70s, and first drugs on the market were in the 90s. The idea is that once you actually try to discover these antibodies that work for a given disease, so they bind to your target, and once you have those, you extract their gene, and then you play around with bacteria, and you teach these bacteria to produce these antibodies based on the genes, right? Very simple manner. And the person that asked me the question of the curve before was an R&D engineer in the manufacturing process. So these kind of processes where you have these kind of factories where you build these antibodies, right? So how does that production work? Just to simplify again, you have two important things that happen. So you have these need to grow these bacteria, right? So you need to make them reproduce, and then at the same time you need to ask them to produce antibodies, right? So they engineer to produce antibodies, and so typically that works as in batches. It's a fermentation process. So same way you produce beer, probably big difference, but in the end it's still a big kitchen. So you start with a very small amount, which is called the inoculum, and that amount you start feeding it with chemicals like glucose, relatively simple stuff. And at the beginning you optimize the feeding so that the bacteria reproduce and grow, right? And you start with a few liters, and it grows to 80 liters, 800 liters in the really big stuff. And then at a given moment, you start switching the feeding strategy, the feeding mix to optimize towards antibody production, right? So once you have, because it's a compromise, a cell, well, these are not cells, the bacteria, either it produces antibodies or it reproduces, this consumes energy, so it has to choose, right? And via the feeding, you can orient what you choose. And so in the end, what is complex about that manufacturing process is that you have to choose the combination of chemicals you feed it, at which moment in time, how much, and then a whole range of other stuff like the temperature, like the turbulence that you put into these containers. These containers are called bioreactors, and again exist in multiple sizes as they move on. And so typically in the lab that guy was working on, these batches last 2 weeks. And he was in the R&D, what they do is experiments. So they continuously try different combinations of these mixes, of these feeding strategies to find out what works well. And it's really important because these drugs are extremely expensive to produce. So these 800 liters that I mentioned, it's mainly bacteria in the end. So you have to throw all of that away, only keep the antibodies that were produced, very, very small quantities, and those you bottle and you ship to patients typically. So what they ideally wanted, their idea was to guide them in terms of which experiments they're going to do. They wanted to be able to simulate this in advance, right? And so their vision was, what if we had an app like this, where on one side you start putting different scenarios of feeds, right? So there you see on the top feed A and feed B, and for every day how much do you put, so, and then the glucose in the bottom, which is kind of the main thing. And so the idea is you can enter those inputs and you would see on the bottom the different metabolites, so the different stuff in the mix and how they evolve. So typically the different curve could show different things, one of them being the antibody. Here, for the purpose of the talk, I had to hide a bit of the stuff, but you get the idea, right? So that R&D engineer knew his biologics, right? Has a vast knowledge about how these processes work. There's a lot of theory involved, and so this simulation model was pretty clear to him. And it was basically, when you think about it, the kind of model is very mechanistic. So once you know everything about your system at the time t, so the different metabolites, but all the other stuff like temperature and so on, it can calculate based on just a few equations how the state's going to be in time t plus 1. Right? So the idea is that you can then start at time 1 and propagate everything until the end, and based on the inputs that you've put, right? And so that model was based on two things. One is you have equations coming from theory. In this case, it's biology and chemistry. There are a few equilibrium that are always correct, and so you can that's part of what the model is built on. But then the other part, because biological theory is not at the stage where it can explain all of this phenomenon just based on first principles. So part of it needs to be shortcut, and those shortcuts are just based on observations, right? So some of these equations are just observed, and this is what we had shown before, right? So actually in the actual model there were 9 kind of relationships that needed to be given to the model, right? And so 9 functions actually, and so these functions, that's the curves we were talking about before, right? So which and it's always the same quantity on the x-axis. It's a certain characteristic, and on the y-axis you have the concentrations of the different metabolites that for which there are these relationships. So still small data problem, but really what that person needed was, can you give me these functions? Can you give me these 9 functions? And actually, can you create a system that generates those functions automatically for me based on new experimental data, right? So the idea is that app, that this person can upload his data to that app, and then that app creates the functions in the back end, generates sends it to the model, and propagates and creates the results, right? So now comes a bit more the technical part, which I think It's finally going to be interesting. So most of the curves look like this, and when you look a bit more into the details of this, indeed, the part at the left is it follows kind of a straight line, right? So linear regression should be fine there. But on the right, it's not like, it's something completely different, right? What really happens is that there's something very unstable on the right. And indeed, when we made that observations, just looking at the data, the R&D engineer told us, yes, of course, it becomes unstable when you reach a certain number, when the value on the x-axis grows. We just don't know how to model it, right? But in the end, that's the interpretation you should do from that big variance that you have there. And all of the other graphs are pretty similar, right? On the right, they have a very high variance. So what we proposed was, let's not use a curve, right? That's quite artificial. What you would like is actually to model the uncertainty in your model, right? At the moment, your model is very mechanistic, so it assumes that you have an exact quantity, but it's not the case. So let's just model that. Do you need to change your mechanistic model? Hopefully not. So that's what we were aiming to do. The idea to do that is then to model this kind of phenomenon using probabilistic programming. So in this most simple idea, also called Monte Carlo simulations, many of you probably have heard of. The idea is that instead of having a function that produces a point value, so point curve, you add some randomness into that, right? So the function is not anymore giving you a single y value. It has its own error. So if you use that function a few times, it will each time produce a slightly different y, right? And the idea is that if you do that, also your model, which uses these functions, will also produce slightly different output every time, right? Which is a great thing, because you can now run that model 1,000 times and see 1,000 different outputs and see the distributions of the outputs, right? Which gives you a good idea of that uncertainty. What uncertainty do you have at the end of your experiment? Which is exactly what the scientists needed to know, because currently they have this impression that there is a precise answer, but actually there's a lot of imprecision which they don't know how they can't quantify, right? So the whole goal of this piece was to quantify that and at the same time model it more accurately. So let's go a bit into code. Let's show a simple example of such a function. This is in R, very basic. You see, can I go to the other thing? Maybe it's easy to see that. Get people moving their head. So you see, very simple. On the left side, you have the equation here, right? That's a very linear equation with two basic parameters and the error here. Error follows a normal distribution. And then once you're on the right, you get to this equation, which is simply a mean and another error. And that error has a much bigger standard deviation. There you see it, right? And so you get this thing. And this looks pretty much like the data we had in the other charts, right? The only thing is that every of the 9 metabolites have a slightly different kind of coefficients on the regression line, and also different variances here and different kind of mean on the right. One of the things we noticed is that it looks like on the 9 charts, the threshold, we don't know where the threshold is, and from experiment to experiment that's a different place, but it's at the same place for the 9 different charts, right? So we also wanted to have a single threshold where that kind of variance explodes, okay? So this non-linearity happens. So this is very basic. If we have that kind of function, again, this function is non-deterministic. You have uncertainty built into this normal distribution, right? And if we give this function to the model, the mechanistic model, it's going to work, right? It's going to be able to run the mechanistic model 1,000 times and get the output. So now the question is just how do you get those parameters? The parameters at the top, right? Can I put here in the middle? Yeah, these things. The A's, the B1, B2, the SD1, SD2, and the breakpoint, right? All of these we need to know. Here, this was just a demo, just to check the concept, but these we now need to be able to estimate, okay? And so that's where another kind of probabilistic programming comes in, which is which actually can, so you can use probabilistic programming to do Monte Carlo simulation like here. The other thing you can do with it is actually estimate these parameters. So in this case, we did this with a program called Stan. I'll show you some links later on if you're interested. So the interesting part about Stan is that you can actually do inference with a language that is very similar to what you saw in R to do the simulation. Again, you see the same thing here. We write the equation of the linear part on the left side, right? We have an alpha, a beta, and so on, and also its own variance. And then on the right side, so if you're on the right of this breakpoint, BPT is the breakpoint. You have another called nu here. You have another mean, and you have another variance. Okay, and it's a Bayesian framework. So the way it works is that the quantities you want to estimate, you initialize them here with a distribution. That is your prior, right? So relatively non-informative. Cauchy is slightly different. Some physicists here will be familiar, but it's we could have taken the normal. It's just much more efficient to do the fitting using a Cauchy. But it's pretty non-informative for our problem. So it's pretty wide distribution. You don't know much where your alpha lies at the beginning, right? And then it's gonna what this system, what Stan's gonna do, is gonna fit this to the data and estimate your posterior. Posterior means everything you know about that variable when you take into account the data, in simple terms. And what you have here, what you notice here also is that this breakpoint, we also estimate it, right? So we don't give it, it's an unknown, it's also gonna be estimated. Another thing you can see is that it's hierarchical. So here we've put actually all of the data of the 9 charts at inside the same estimation routine, and we have actually a different alpha for every metabolite. Metap is the different metabolite, so the 9 different ones, right? And so a given data point belongs to one of the 9 metabolites, and each of them has its own alpha and beta, right? So in the end, these are independent, but the breakpoint is common to all of the 9. So that's again a cool thing about this method is that you can really engineer your model, make it hierarchical, create these kind of breakpoints. You can do all of that, and still get your parameters. Have a look at an output here. Again, using an interface from R to generate this, and you see results here. Again, no p-values here and results. So Bayesian scheme. So what you see is actually what Stan allows you to do is not to estimate the values. It allows you to sample from the posterior. And then what you do is you just get means and so on. So this is the mean value and standard deviation. So it gives you confidence intervals on what you know about these variables. So here you see the different alphas for the 9 metabolites, the betas, the nus on the right. And if you plot this just to check what it means, you can look at the mean is on the red line here, and then the blue thing shows you the kind of how it's distributed, how the samples are distributed if you sample from it. So you see it estimates the breakpoint, it makes sense, it looks like the graph and so on. Right? Green you can ignore it. It's from other stuff. So that's the idea of this this thing. Now the goal of all of that was to get the results of the mechanistic model, right? And so here they are, well, on two metabolites. So now you see here the propagation. These are the 14 days I mentioned, the 2 weeks each time. This is each time one of the metabolites. See, this is an interesting quantity which grows over time. And now you see you have a quantification here of the uncertainty, right? So we have propagated the uncertainty from the woo. This is dead. Well, I would like to show the next slide. So let me use that. I think this is going to be more efficient. Two more slides. One is conclusion of this tool. Why I like this so much. One, thank you very much, Thomas. Yes, I think so. Yes. So one thing that is, oh, that's just, yeah, that's nice. One is really cool that you can model the data generating process, right? So in the end, it's a generative model, right? So you can fit those parameters the way you want. You can really engineer it. And think about, otherwise, how would you do it, right? In R, if you look for a regression that is a bit special, you might find a segmented regression. You might find a splines regression. You might find some hierarchical models of one level, right? Random effects and stuff like that. But if you really want to fine-tune exactly your model the way you want it, the way that really makes sense in terms of the observations and in terms of the logic behind, you'll never find exactly what you need. So here the basic idea of you have a regression, you have a line, and then a variance that explodes. Will you find an implementation? Of course, you can make your own, and if you can work with likelihoods and so on, that's great. But this kind of framework allows you to do that in a very easy and expressive manner. And then the second thing is, once you have this, you can propagate the uncertainty through a mechanistic model. So when you interact with scientists who have their mechanistic knowledge from their domain, it's a very useful tool to collaborate. So I didn't put the name of the discipline, but basically the part at the bottom is what's called Monte Carlo simulation, and the part at the top is called Markov chain Monte Carlo simulations, which is the process of estimating posteriors using the Bayesian approach. And then finally, if you're interested in those kind of tools, JAGS and STAN are two specialized tools based on C, so very efficient also to do this kind of fitting. You have an implementation in Python, PyMC3, which is pretty good too. And then very new, you have on even a probabilistic programming framework based on TensorFlow now, which is very new. I've not tried it out. The promise is that it can leverage GPUs, of course, to do that kind of stuff. But it's certainly an exciting piece to look at. Yeah. And then finally, what gets you excited is applying this kind of problem to healthcare. We're always looking for passionate people about this. So you could look at our website, search for data science or artificial intelligence. There are a few jobs open there. So that's it. Welcome any questions, of course, or later at the bar.
Thank you. Questions? Yeah.
Thanks for the nice presentation. I have just a question. In your functions graph, all the functions graph which you have presented,
Yes.
one of those functions graph was like F8 or something. In that there was this huge variance in terms of at the breakpoint. There's like all these points are going all around the graph. So in terms of test, have you like, how do you interpret those tests? And because if you're modeling that kind of such a huge spread, does that really make sense? So yeah,
I'm not sure I understand the question.
so if you go to your functions, like so if you see F8, there is a spread over all at the breakpoint. So how do you make sense of this one?
At the right of the breakpoint, you mean?
Yeah, at the breakpoint.
Indeed. So,
Thank you.
by the way, there's one exception. Function 4 has its breakpoint on the left. Actually, we had to exclude it from the previous routine. But that's a detail I want to simplify here. So function 8, I mean, all the functions have the variance that explodes after the breakpoint, right? And we don't know much what's happened, what that distribution looks like, right? You can't really say it's a normal or whatever, but we don't really have many hypotheses about what else it could be. So we didn't do too much theory, frankly, right? What we wanted was really to have something that is more or less similar to the actual phenomenon, right? So that's this idea here, right? So what we do is try to model it, try to make sure it looks more or less the same. You could formally go a bit deeper in terms of understanding, you know, whether this kind of distribution is distinguishable from the original ones. But no, it's not an area we went deeper. It's not that's going to really change the value of the exercise. So not sure I answered the question, but.
Hi, thanks for the nice presentation. I was talking this week to a keen proponent of Bayesian analyses, and he mentioned that there's no such thing as a non-informative prior. So I was wondering if you could motivate how you chose your priors and if your results are sensitive to the choice of prior. Thanks.
So disclosure, I'm no statistician, right? I skipped all statisticians course at math class. Some of you know it here. So I'm a very pragmatic guy. I took the prior there from Gallman, so reference statistician who talks a lot about this, explicitly gives good guidelines about how to choose your priors. The idea is, I think here it's more via trial and error that you can see whether it changes anything to make it even less informative. Of course, it's always slightly informative. There's a structure that is quite fixed, and then you have to put a value, and if you put it too big, it's not going to converge. So yeah, there are some choices put there, but the idea is if you change those values by something reasonable, it's not changing anything on the conclusion in the end, right? So very pragmatic at this stage here. Yeah.
Last question. You want?
Okay, thank you for the talk. So for about this model, so in the first range on the left, so you have like basically Bayesian linear regression. So actually, your uncertainty on the left will be kind of the same as in the middle, probably. But when you see on the graph that on this function, so you see that it seems that there is also some kind of uncertainty, like when the value on the x-axis is near to 0. So I was wondering, like, I think maybe like Gaussian processes could also kind of capture this effect. So I wanted to know if you use Gaussian processes, and if and if you did, like, what did it give?
Yeah, it's a good idea. I didn't do it.
Because it seems that really on the left you have like also this uncertainty, while really on the middle you don't have it.
Yeah.
So that's something maybe you would like to capture.
That's a very good point. Maybe we're over-interpreting though here, right? I don't know. There are definitely some curves like F9, right? Where it looks again to confirm that. Maybe it's just because we have more data points on the left. I don't know. You could also extend this model, right? You could actually make the variance a function of your x-axis. If you can try to find some trend there. Again, the thing is you don't want to over-engineer too much here. There's really not that many data points, so the less moving parts we can have, the better.
Yeah. So, but since Gaussian processes don't have as many data points, I mean, you just have to design the kernel. So probably it could be a good fit.
Yeah, good idea. Thank you.
Thank you, Nassim, again.
You're welcome. Thank you very much.
So the last talk after mid-sized company, a startup coming from Louvain-la-Neuve, and so the floor is yours. Thibaut, you're switching the mic, or you want this one?
So good evening, everyone. Two very interesting talks already. So indeed, I have just prepared two or three slides just to present the analytics, which is a much smaller company than UCB, of course. So you might have never heard about it. So we are only active in healthcare since 2012. For myself, I have a PhD in machine learning, and I apply such techniques in healthcare for about 15 years now. And the ambition of our company is to improve the design, discovery, and deployment of new healthcare products and services through data science. And we are active in mainly 3 different fields of applications. All the examples that you see that are underlined in yellow, orange are clinical decision support applications. In purple, it's public health policy making. And I will give an example later in the talk. The gray part is more related to the two previous talks that we just had. So in drug discovery, we develop and maintain a software that is, to make it short, a bit between what we have seen in the first presentation and what you would find within IBM Watson for genomics, for example. So it relates genes to proteins to targets of drugs. And we are also active in drug manufacturing, where we gather the data from production line in biomanufacturing plants, so just like the previous talk, mainly to discover what are the main causes, main parameters driving the yield of such processes, or to detect problems while producing the batches. And in orange, I will just give an example. For example, you see here on the top right, treatment response prediction in the context of allergy. So for example, in that case, we help a company called DBV Technologies in Paris and New York. They develop a patch to desensitize children from peanut allergies. Nowadays, the only way to for the clinicians to assess that the child is actually desensitized to peanut protein is to perform what is called a food challenge. So you actually give the peanut protein to the child in very small amounts, up to the point where nothing happens, hopefully, or to the point where the child reacts. But still nowadays there are a few deaths every year because of these food challenges. And what we have done for that company, for example, is to identify proteins in blood of these children that allow us to compute a model that predicts the results of the food challenge. So instead of directly performing this food challenge, now the clinicians can have a blood sample from the children, run our model, and the model tells the probability that the food challenge is safe to be performed or not. So and you have other examples in cardiology, urology, clinical trials management, and so on and so on. And indeed, in this case, we are used to deal with very different types of data, DNA sequencing, RNA sequencing, psychological traits of the patients, more traditional clinical data, etc. And most of the time we combine different data sources into the same predictive questions. Of course, what is very typical of data analysis in healthcare is that there is no such thing as big data. We just touched upon this question in the previous talk. Even when we talk about phase 3 trials, we'll have only a few thousands of patients recruited if you're lucky, but the number of parameters that you will measure on these patients will explode to several tens of thousands or even millions or billions of different features. So you have not a big data matrix, but a fat data matrix. And in that context, it's very, very difficult to learn predictive models that will be robust when you try to apply them on future patients. And even if you consider biomanufacturing, as in the previous talk, you will typically have hundreds of different features for raw materials, quality control results, certificate of analysis, process data, or even within the reactors or fermenters, you will have one measure every minute or every 5 minutes, and when you combine all these features, it's always much bigger than the number of points that you have. And so at the analytics, we try to develop several tools and frameworks for analysis of this kind of data. We heavily rely on R libraries, namely the tidyverse packages, but we are also developers and maintainers of our own codes. Or base codes we call it bricks, but we are and this we keep it private to the company. But other pieces of software we maintain and we release them as open source software, namely the liblinear package, for example. So it's SVM, especially fit for very large matrices. We are the maintainer of that in the CRAN repository. What I will talk about now is a specific product that we developed. So we have, in fact, in the company 2 activities. One is basically consulting. But another one is to develop a real in vitro diagnostic platform, so a real healthcare product that we develop and commercialize, which is called the RheumaKit. So it's a platform in rheumatology. Through that platform, our goal is to provide rheumatologists, so it's a platform for health professionals. We aim at providing them with both molecular and data science insights to better manage their patients. And so through that, we aim at supporting them in their practice, but also, of course, to improve patients' health. And you will see that it has also a link with the sustainability of the health economics of our healthcare systems. So I don't know if you know about arthritis in general, but it's a set of diseases, a family of diseases that is highly prevalent. So we speak about 1% of the worldwide population that has some form of arthritis, which is much higher than cancer, for example. And it's a heterogeneous set of diseases. You have different forms of arthritis. Osteoarthritis is more a disease of aging. The joints get used and degrades over time. So this is rather correlated with the age of the patient. But you have other forms, like rheumatoid arthritis, which is an immune disease where the body degrades its own joints. And this is not a disease of the old population. You have juvenile arthritis, and you have many patients in their 30s developing rheumatoid arthritis. You have other forms, psoriatic arthritis, microcrystalline arthritis, which is more familiarly called gout, for people eating too much sugar or drinking too much alcohol, for example. And so we identified two different challenges. The first one is the fact that for a small proportion of the patients, the differential diagnosis between all these different forms of arthritis is very complex, and that is what we call undifferentiated arthritis. So in those limited number of cases, it's difficult for the rheumatologist, simply based on symptoms, to distinguish between an osteoarthritis, a rheumatoid arthritis, etc. Another challenge is to prescribe the right drug to the right patient, in particular in rheumatoid arthritis. So once you are diagnosed with rheumatoid arthritis, you are prescribed with a synthetic drug, so a small molecule, like we saw in the previous presentation. Not the same, but also a small molecule, which is quite inexpensive and will work on about 60% of these patients. For the other 40 patients, you get to a second line of treatment, which are biologics, like the big molecule we saw on the right of the slide of the previous talk. And you have, in fact, more or less 10 different mechanisms of actions of such drugs. You have, well, I will describe them. Each of these mechanisms is effective also on more or less 60% of the patients. But you cannot tell in advance which drug will work on which patient. And if you see the official treatment guidelines in the US and in Europe, you see some form of trial and error in the bottom left of the guidelines. So you have to pick a drug at random among these families of drug, prescribe it, wait for a few months, and then if it doesn't work, you choose another. As long as you have not identified the right drug, the joints degrades and degrades. There are also painful effects, side effects of these drugs, so better not lose time prescribing not the right drug. And it's also a very costful set of drugs. We talk about between 10,000 and 15,000 euro per patient and per year. And since these are chronic diseases, it means that you continue the treatment lifelong. Just to give an example, there are different big families of such drugs: anti-TNFs, anti-IL-6, JAK-STAT inhibitors, T-cells inhibitors, B-cells inhibitors. And in total, in Belgium, these drugs represent 10% of the global healthcare insurance budget, just for rheumatoid arthritis. So this set of drugs, just for one disease, takes 10% of the budget, and it's highly inefficient in the way it is prescribed. So we can first, from a health economic point of view, and that is where we are jumping into data analysis, we can ask several questions about the way these drugs are prescribed. So we made an online app based on the Shiny framework. You have the URL here on the screen. You can play with it. I just made a few screenshots. So this application combines 5 different publicly available databases. So the TARDIS databases, at least the public part of the TARDIS databases, which is a database where each rheumatologist in Belgium has to encode every single prescription of such drugs. We combine to that database the Belgian census data, the IGN map of Belgium, bpost for the correspondence between zip codes and provinces, and the database that's available within INAMI, RISIF, for the rheumatologist locations in Belgium. First observation, so these are actual prescriptions market shares of these different drugs in Belgium. And they are grouped more or less by family. So all the shades of yellow are anti-TNFs. And you see that there is a huge predominance of these drugs with respect to other mechanisms. And this has no reason to be. They are not more effective than other families of drugs. It's simply because they are the first that have been introduced on the market. So just by habit, rheumatologists prescribe them. And you see that other types of drugs like anti-IL-6, which are much more recent, are pictured in red. They are they are in brown. We almost do not see them on the graph. So there is an unbalanced market share that can be witnessed in the data. You have also other strange observations. For example, you see the prescription of such drugs per thousands inhabitants per province. And if you click on the Limburg province, for example, or West Vlaanderen province, you see a rate of prescription almost 2 times higher than in Namur. There is no reasons for that behavior to be observed, because the prevalence of the disease is not that higher in Limburg than in Namur, for example. So, of course, based on this kind of analysis, and on the real app, you can also play to see how these prescription rates evolve through time. Based on this, we are also discussing with INAMI and other mutualities to get broader access to the full databases to analyze more finely what happens there. Now, that was more for the health economic part. The RheumaKit platform itself, it's a combination of different tools. There is 2 different molecular tests combined to data science analysis, one for differential diagnosis, the other one for treatment guidance. The one for differential analysis is already CE marked and thus used in some hospitals in Belgium and in Spain. The other one is still under a clinical evaluation within a clinical study. These tools are operated as a diagnosis as a service, meaning that the rheumatologists harvest the sample on their patients, they ship it to a central lab, which is one of our subcontractors, to which we specify which markers we wanted to measure in the patient samples, and I will get to how we identify these in a minute. And then the data are uploaded on the platform, and the data are interpreted, and the report is delivered to the rheumatologists. What you see on the right is a clinical follow-up application that is freely available. It is the way through which we deliver the results of our diagnosis, but it is also a way for the rheumatologist to capture other forms of disease activity scores or diagnostic scores that we did not invent, but that we implemented. And this part of the application is now used by more than 40,000 users per year over the world. And we are currently integrating these applications with the Belgian electronic health record. So I don't know if you know the Réseau Santé Wallon, which is the French-speaking part, electronic health records. In Flanders, there are 3 different ones. The most known one is COSO, but anyway, all these frameworks are interoperable. so that rheumatologists will not have to encode the data in two different places. And there is finally an app for the patient so that they can record their easiness or difficulties to perform everyday tasks, and if they want, share that information with the rheumatologist, who then sees an extra curve on the web application. So how did we come with this molecular diagnostic solution? We started by recruiting patients for which the diagnosis was clinically very easy to perform. So no undifferentiated cases, but clearly differentiated types of arthritis, rheumatoid arthritis, lupus, which is another immune disease, osteoarthritis, spondylarthropathy, microcrystalline arthritis. And we analyzed the expression, so the activity of more than 50,000 genes. And from these data set, we identified a set of more or less 100 genes that we wanted to validate in a further clinical investigation. If you just stop there, you have a high risk that either your gene selection or your model will overfit the data. And a second item is that these were very clearly differentiated patients, so we haven't made our point yet. But we believe that if we started with very differentiated patients, the identification of the genes discriminating these different conditions will be easier to identify. So what we have done next was to recruit another set of patients, about 100 patients, for which we kept the 100 genes that we identified in the previous iteration, but this time the aim was not to select the genes, but rather to build a predictive model based on these genes. And most of these patients there were undifferentiated arthritis patients. And that poses a problem of clinical design, because you have to recruit these patients at a point where they are in that state of undifferentiation of their arthritis, but then you also have to prevent from treating them, because if you treat them, you bias their clinical evolution. And that explains why it is very difficult to have a much larger cohort of patients, because it's also from an ethical point of view, it's very difficult to say, okay, thank you for participating to our study. To reward you, we will not treat you for years. Because the average of the delay between the point where we want to make the diagnosis and where we recruit these patients and the point where they naturally become differentiated is, the median is 3 years, and some of them have been followed for more than 5 years. What we observed in that step was that there was clearly a predictive ability of our model solely on the transcriptomic signature, so based on these 100 markers, but that is predictive powers were improved if we also add more traditional clinical descriptors of the patient. So we also, with feature selection techniques, we identified 3 clinical parameters among 12 that were suspected by the clinicians to play a role in that diagnosis. So we ended up with a bit less than 100 genes and 3 clinical factors, and we came to a final solution. But at that point, we have another challenge, is that you want also, you do not use the same measurement techniques when you measure 50,000 genes expressions than what you want to measure in a clinical routine setting. The technology that is used to measure that is either microarrays or RNA sequencing, which is quite expensive from a research point of view, and in the clinics you can at most have quantitative PCR, which is already expensive but more reproducible, but which is more fit to target a smaller set of genes. So we still have another change to introduce, is that we keep these genes and we keep these clinical parameters, but now we have to build a model on a qPCR platform and then to finally validate that final solution on an additional set of 31 patients in that objective. So that's what we did with satisfactory predictive performances at the end of the road, and then now that is how the diagnosis part looks like on the bottom left of the screen. So the rheumatologist specifies the 3 clinical observations, transcriptomic data are uploaded by the lab, and the model computes the most likely diagnosis, hiding behind this state of undifferentiated arthritis. There is also the perspective of being able, of course, to accumulate data over time, because now the solution is in the clinic, and now each time a patient benefits from the diagnosis, we have an extra row in our matrix. And if we have good contacts with the rheumatologist, we could or we can have a feedback on the fact that our model actually made a good prediction or not. And so there is the perspective of improving these models over time. But there is a limitation, which is a regulatory limitation. You have to see a predictive model in a clinical context just as a drug or another diagnostic or medical device. So, which means that its performances have to be evaluated within a clinical trial. So if you change the parameters of your predictive model, you are actually changing your solution. So in practice, what we do is that we still monitor the progresses that we could make by accumulating the data, but we do not automatically update the model. We do that every once and then we also update the technical documentation associated to our product. Just a hint on how this model works. We tried a lot of different models. Finally, the solution that performs best in this setting is a k-nearest neighbor, which is quite simple. We pay attention to the fact that k must be sufficiently high so that the model does not exhibit too complex decision boundaries, so it's smoothened by the fact that k is rather high. And the distance metrics used in that model is a mix of a distance computed on the clinical parameters and the distance computed on the transcriptomic parameters, with a trade-off parameter between both components of that distance. A small trick, although the lab tries to put the same amount of RNA in each PCR experiment, you cannot guarantee that it's always fully reproducible, because measuring that is also complex. So we designed a distance that is based on a correlation measure, meaning that if you have a global shift in the measurement of those genes for a given patient, which will be triggered by an increase of the amount you put in the PCR, then the distance is not affected by that. So we are robust to variations of the amount of RNA that is input in the process. On the other hand, if for a given patient you will have different level of expressions of different genes, then the distance will be affected. In general, we heavily make use of techniques for feature selection that are more robust, because we operate in that context where we have many features and a limited set of samples. A very easy trick you can implement is to use ensemble methods. So I don't know if some of you know feature selection methods. The easiest one is you perform a t-test on every feature that you have, and you rank your features by increasing value of p-value. But instead of doing that on your whole cohort of patients or on your whole set of observation, you take subsamples of this data set and you compute various rankings, and then you take the consensus ranking of the importance of your features. Then you select only the head of that ranking. So that is algorithmically very simple. It appears that it works well in practice. Another way to do, which is more mathematical, more elegant, but also a bit more complex, is to heavily use on regularized models. So that is what is pictured here. You do not have to fully understand the equation, but basically the term on the left is stating to the optimization problem that you want to classify your patients correctly, you want to make good predictions, and the parameter on the right hand just states in that case that you want non-complex models, so simple models. So the weight of the models are penalized, and you can impose extra constraints. So for this example, what is imposed is that you will select only groups of features, in this case groups of genes, that are predefined in full or not at all. So if you know that some genes operate within a given metabolic function together, you impose to the model to select all of them or none of them. You can play with that framework with very different regularizers. You have L2 regularizers, L1, which will produce more sparse models, TV norm, which is more used in image processing. PSFS, so partially supervised feature selection, is another form of such regularizer when you can encode prior knowledge that you will get from medical experts of biology experts. So we can speak about that for hours. The other indication of the test is how to help rheumatologists to prescribe the right drug to the right patient. What we already deliver with the first version of the test, before we get to a more advanced one, is that we observe that within the 100 genes that we have selected in this diagnosis test, most of them are related to target of existing drugs on the market. That has never been told to the feeder selection model that we should favor these or these genes in this case, but it happens that about half of these 100 markers are target of existing drugs. And so what we do is to provide the rheumatologist with a way to quantify the activity not of a single gene, but of a metabolic function for each patient with respect to a reference cohort. So what you see with this particular patient, so this represents one patient. What you see, for example, is that the TNF pathway for these patients is already very low. There is a low activity of the TNF pathway. So better than choose your drug at random, we recommend to choose another mechanism than anti-TNF for this patient. Sometimes it's the opposite. But for example, you have the JAK-STAT pathways, which is the target of another available drug, which is very high in brown here. So why not, better than choose purely at random, try that one before. Of course, one gene expression is not numerically comparable to another one. They live in different ranges of expression naturally. So we have to implement a way so that we can compare these different pathways. That is how we do that. What you see at step 1 is that we position a single patient with respect to a single drug mechanism with respect to a reference population. For example, in this case, you see a patient which, for one given drug mechanism, drug target, is situated at the 90 percentile of a reference population. So we translate that in the lower left panel by displaying a 90% value for that mechanism for that patient. And then we do that for different drug mechanisms. There is a whole set of standardization details that I skip here. And we can do that for many different patients. And this is what is available for the rheumatologist on a patient-per-patient basis. What we keep private for the moment is the meta-analysis of that, being what can we say at the population level? Are there some mechanisms, some drug targets that are predominant in a larger part of the population, or is it more equivalent for all drug mechanisms? So that is a research field in which we are active on our side for the moment. And that's it. So if you have questions, I would be glad to answer them. Thank you.
Thank you. Any questions?
Thanks very much for your presentation. I had a question regarding sample sizes. I was wondering, some models were built on 100 cases, for example. How do you know whether this is somehow representative of a larger population, or how do you somehow take into account power calculations or something?
So as we conduct prospective studies on a fixed solution, so just to test it, on that population we can compute an average performance, and we can also, by subsampling analysis, compute a confidence interval. on this performance. And if you see the notice of that IVD product, these are reported. So we are fully transparent about that. Of course, we would like them to be smaller, but it's like that. It will improve over time. We are transparent about them. It's still better than the purely clinical, because we also compared on these patients with respect to standard practice. We are clearly in a different range of performance than the standard way to diagnose these difficult patients. Confidence intervals are quite large, but we communicate them. Yes?
You said that your product was validated. Is it in like?
Sorry?
Is it commercialized currently, or you are still under trial? What are you currently doing?
One version is already commercially available. If you go here in Brussels, we have two hospitals providing it, so Saint-Luc and Saint-Pierre. You can find it in different hospitals in Ghent, Leuven, and Liège. Another version, which will really predict the response to different families of treatments, is under clinical evaluation.
And have you calculated the predicted values of your kit?
Yes.
Like, and what do they look like?
It depends from pathology to pathology, but this is available online. If you go to rumakit.com, you will find the precise information in terms of sensitivity, specificity, and predictive values as well. In terms of balance classification rate between rheumatoid arthritis and other forms of disease, we are at about 90% of balance classification rate.
Okay, thank you very much. I think we can go to the ring as we have are a little bit behind schedule. Thank you again, Thibaut.
Thank you.
Just a last word from Philippe.
Okay, well, it's been nice to see the data science community in this room. Last time we were upstairs. This is a room which can still hold another 50 or 100 data scientists more. So talk about it. If you like the evening, talk about it and make sure that we are more than 100 next time. There is something important that you have to book. It's December the 20th. December the 20th, we will have our first anniversary of this place, and just before that, starting at 4, we will have a job fair for data scientists. So data science boot camper finish their training, and they become available on the market. So there is a data science boot camp on the 20th of December, followed by a big celebration, of course, because that's what we like to do. About celebration, the bar is open, and I hope to see you all here next time. Thank you very much. Thank you for the organizers. If you have ideas on topics that can be showed, go to the guys that are organizing it.